Meet the springtail, my tiny co-host for this episode. Springtails are creatures that live in soil all over the world. Seriously, they're everywhere. They can be found on every continent, in every country, and in almost every patch of soil. Because there are so many of them, springtails are perfect for studying how species survive and adapt. We'll find out more about them in a little while when we visit the Baumla Lab at Georgia Tech. But first, we need to talk about genetic variation. Genetic variation is what makes individuals in a population slightly different from one another. It's why some people have brown eyes while others have blue, or why some plants can handle drought better than others. This variation helps species survive changes in the environment, like disease or temperature, and it's happening all around us. You've likely seen it without even realizing. Do you put on sunscreen when you go to the pool or the beach? Well, that's to protect from ultraviolet light, which can cause mutations. Mutations are changes in DNA, and they're a major source of genetic variation. But not all mutations are the same. Some mutations can affect just one gene. These are called gene mutations. These might change just a single protein, which can lead to changes in one specific trait, like eye color mentioned earlier. Other mutations affect larger sections of DNA, or even entire chromosomes. And because they involve many genes, they can impact several traits at once. It's important to remember that not all mutations are harmful. Some are neutral, and others can even be helpful. If a mutation gives an organism an advantage, like resisting a disease or surviving in a new climate, it might get passed on to future generations. That's how species adapt and evolve over time. To understand where genetic variation comes from, we need to look at two important cellular processes: mitosis and meiosis. Let's head to Georgia Tech's Baumla Lab and University of Georgia College of Veterinary Medicine to explore how these processes work.
Genetic variation refers to the differences in DNA within individuals of a population, or the genetic recombination that occurs across time in that population.
Genetic variation simply means that if you look at a population of organisms, they don't all have the exact same gene sequences, that there's variation in the genes between any two individuals.
Mitosis is when you have two cells that split into two diploid cells. This is basically when you have the chromosomes realign and then separate, forming two cells. And this is important for growth, and this is important for asexual reproduction.
So meiosis is basically the process by which DNA is replicated and then divided into daughter cells that are the germline cells, either sperm cells or egg cells. And there's a couple ways in which meiosis can lead to genetic variation. One is the fact that each of our cells has a pair of each chromosomes, and one that we got from our mother and one we got from our father.
For genetic variation, meiosis is really important. It allows for you to have these 4 different cells that have different recombinations of their chromosomes across the space. You can have substitutions or deletions, and that can make micro changes in the actual genetic variation. And those can introduce different levels of mutations that can have some sort of phenotypic response. During meiosis, when you have these sorts of crossover events of your chromosomes, you can lead to these sorts of broader differences in the chromosomes and broader differences in genetic variation between your different haploid cells. For inheritable genetic variation, it needs to be passed into those haploid cells, those gametes that are going to be passed down to the next generation. And so any mutations that occur in cells that are just going through normal mitosis, those won't necessarily make their way into the next generation. Errors during DNA replication can come from point mutations, substitutions, deletions. All these sorts of things can affect the overall code that builds our DNA.
Errors that can occur during DNA replication is, for one, the DNA polymerase that's copying a strand of DNA is supposed to put in an A and puts in a G instead, for example. Or it may make a mistake and put in 2 nucleotides when it should have put in 1, or misses a nucleotide.
Non-disjunction is an error during the meiosis process where the chromosomes fail to separate. And you can have, depending on which side of these haploid cells, these gametes, differing numbers of chromosomes within that cell. So this can lead to offspring that have different numbers of chromosomes that can have huge impacts on their development. Genetic variation is really critical because it allows for populations to be very resilient to environmental changes over time. A lot of environmental factors can impact or increase mutation rates in species and organisms, and this can be temperature, this can be UV, these can be chemicals that are released or put into the actual environment. All of these things can have an effect on the ability or the increased rates of mutation in these organisms. Springtails are a small hexapod. They're actually pre-insect. They diverged from the insect evolutionary lineage before insects came about. Because you can find springtails all over the world, they make an ideal system to study genetic variation and evolution. Here in the Baumol Lab, when we're working on springtails, we often see variations in populations as well as the individuals within them, and that can relate both to temperature and the runoff that can come from streams or chemicals. And so all of these things can have impacts on the individuals and the mutations that occur in populations in those areas.
The study of genetic variation in pathogens, be they the malaria parasite or bacteria or viruses, is really an ongoing thing, and a lot of it is focused on trying to stay ahead of these pathogens. Every year we have to get a flu vaccine because the flu virus has changed. It has ability to change those proteins that the immune system sees, and so the immune system's constantly playing catch up, trying to learn all these different variants of this protein.
It might still be tricky to see how this process connects to our everyday lives, but there are examples all around us, like the way many adults can drink milk. Most mammals stop producing the enzyme lactase, which helps digest milk sugar called lactose, after they no longer require milk from their mother. But some humans have a tiny change in just one gene, a type of gene mutation called a point mutation, that allows them to keep making lactase into adulthood. This small change affects just one part of the DNA code, but it makes a big difference. That's why some people can drink milk as adults without any problems, while others might feel sick from it. So if you're a milk drinker, you can thank genetic variation and a helpful little gene mutation for that. Until next time, stay curious and keep asking questions.